गुड इवनिंग फ्रेंड, वेलकम टू अवर चैनल एग्जामपुर आईएएस इंग्लिश मीडियम। आप सभी का हमारे परिवार एग्जामपुर में स्वागत है। सो वेलकम मृदुल, वेलकम ऑसम क्वीन। You are not attending my classes for last 2 days. What's the reason behind it? And apart from that, try to attend my current affair classes, because for UPSC current affair is the backbone. You have to regularly update with the current affairs. Plus, I am taking 4 to 5 current affairs slowly and gradually. If the student increases, I will also increase the current affair. I am covering all the dimensions. If you are taking any topic, all the historicals, way forward, suggestions, objective, all the things are being taken care of. So try to attend my lectures of current affair also. The schedule time is 7 p.m. Is it okay? And how are you, Mridul? Awesome Queen, कैसे हैं आप सभी लोग? How are you? How was your Independence Day? Are you there in the classes? I'm not getting your comments. Mridul, Awesome Queen, are you there in the classes? Kindly comment. Are you there in my class? Mridul, Awesome Queen, so that we can start our session. Otherwise, if the initials, if the beginnings are missed, then it is very difficult to correlate. I do not. So just post a comment so that I can acknowledge that our students are present in the classes. I'm fine, sir. Okay, I'm fine too. Awesome Queen, are you there in my class? Just post a comment so that I can acknowledge whether you guys are there in the classes or not. Chaliye. So without wasting time, let us start the session. So we have to discuss a stem cell. ठीक है ना? So in this session we will discuss the stem cells. What are its regulations? We are going to discuss the stem cell from the perspective of mains, because it's a mains topic. We will discuss each and everything from the perspective of mains. So the topic is, recently a US patient became the first woman reported to be cured of HIV after stem cell transplant. So you can see that this is the first case in which a woman who was suffering from AIDS has been treated of stem cell therapy. So basically in AIDS, what happened? Now we have to see what is AIDS. So AIDS is what? Acquired immunodeficiency syndrome. AIDS is what? AIDS is acquired immunodeficiency syndrome. So basically what happens, if you see, I will just correlate AIDS with our immune system, because in AIDS what happens? Acquired immunodeficiency system. Your immune system gets very weakened. Your immune system gets very weakened. So now suppose this is a virus. And this is the surface protein that is present. Okay, so this surface protein, we call it as antigen. Okay, when it enters into a body, a type of WBC cell called monocyte, a type of WBC cell called monocyte. So this is the monocytic cell. Monocyte cell. Now what will happen when this virus penetrates into a body? Just listen to my word carefully. Welcome Jaima Bharti. When this virus, in the topic we are discussing regarding the stem cell, the context is US woman has been treated of HIV using stem cell therapy. So first of all, we should know what is AIDS and how we can treat it. After that, we are going to study the related stuff. So you can see that this is the virus, and over the surface of the virus, these special proteins are present. So this is called antigen. When it penetrates into our body, the first thing that it encounters is monocytic cells. The monocytic cell क्या करता है? The responsibility of the monocytic cell is to engulf this virus. So what it will do? It will engulf this virus. निकल जाएगा. It will engulf this virus, and after engulfing, what will happen? The surface protein that is present, it will get attached to the surface of the monocytic cell. Okay. Now what happens in the blood? T cells, or we call it CD4 cells. We generally call it T cells, but in the case of AIDS, we call it CD4 cells. Lots of CD4 cells are present. It's our intelligence officers that are continuously moving here and there in the blood. So this is called CD4 cells. CD4 cells. Now, what is the responsibility of the CD4 cells? I call it as intelligence officer. The purpose of the CD4 cells is to check whether certain antigens have been entered in your body. Are you getting my point? The purpose of CD4 cell is to check whether certain antigens have penetrated in the body. If CD4 cells found that the antigens has penetrated into your body, now what the CD4 will do? What the CD4 cell will do? It will try to communicate. It will communicate with our immune system. Okay, with your immune system. So it will communicate to a T lymphocytic system. Apart from that, B lymphocytic system. And after that, what will happen? When our immune system recognizes that the antigens of foreign particles have penetrated into your body, this will get activated, stimulated, and after a certain delay of the time, it will release the antibody. Okay, it will release the antibody. This is how our immune system works. And once our immune system releases the antibody, slowly and gradually it develops immunological memory. What is immunological memory? The purpose of immunological memory is to retain. It retains the information, or it captures the genetic information. It remembers that it has released antibody towards the particular antigen. Yes, we are discussing. हम सारी चीजों को discuss करेंगे. Don't worry. So this is how our immune system works. And once it releases the antibody, what happens? Development of immunological memory takes place. This is what? Development of immunological memory. Immunological memory takes place. Now, what is the purpose of immunological memory? It retains the information that it has released antibody to a particular antigen. ठीक है ना? It retains the genetic information. It retains the genetic information of antigen, and it remembers that, it has released antibody. Okay, it has released antibody. Okay, so this is how our immune system works. So you have seen that what is the importance of CD4 cell? The purpose or the importance of CD4 cell is that what? The CD4 cell is a communicating link. Our CD4 cell is a bridge and between monocytic cell and our immune system. Okay, see, the responsibility of the CD4 cell is to recognize whether our body is being infected with antigen or not. And if it encounters the antigen, it will directly communicate to our immune system कि भैया get ready, get stimulated, get ready. Our body has been penetrated with the antigen. So this is a communication link. Suppose now what will happen if this communication link break? So AIDS virus, when you talk about the AIDS virus, what will happen? AIDS virus directly attack the CD4 cells. कितना चालाक है? Just think of it. How much intellectual is the AIDS virus? It attacks your CD4 cell. ना रहेगा बांस, ना बजेगा बांसुरी. The AIDS virus knows it very well that CD4 cell is responsible for maintaining our immunity, because it is the CD4 cell itself which communicate to our immune system. If the CD4 cells are being damaged, then the communication network will get damaged. So AIDS virus क्या करता है? In normal human being, approximately greater than 1500 numbers of CD4 cells are being found. But if he is infected with AIDS, the number will decrease to 200. So now the CD4 cells number will decrease. If the CD4 cells number will decrease, what will happen? Though our immune system will be present, but it will not be working. Because why? Because it is not getting the information regarding what type of antigens have penetrated into a body. So it will be like a sleeper cell. So this is how our immune system work, and you have known that in AIDS particularly the CD4 cells number counts the CD4 cells number count will decrease because the AIDS virus is directly impacting our CD4 cells. Acha, please do let me know what is the difference between HIV and AIDS. Anyone out of you? So you have seen that CD4 numbers are decreasing because when we suffer from AIDS. So resultant आप लिख सकते हैं, you can write it like the number of CD4 cells will decrease. Number of CD4 cells decreases when our number of CD4 cells decreases when people suffer from AIDS, and hence their immune system gets weakened, and hence their immune system gets weakened. Okay, is it clear? So please do let me know, Jema Bharti, awesome queen, Mridul Rajji, please do let me know what. So AIDS you have understood. In basically AIDS, the number of CD4 cells decreases because the virus is directly attacking the CD4 cells. In normal humans, it will be like 1500, more than 1500 numbers of CD4 cells are being found. But when the person is been suffering from AIDS, this number count decreases. It will be like 200. So number of CD4 cells decreases. That means the communication link is not being properly maintained. If somebody will suffer from any type of virus, bacteria, the same information cannot be communicated to the immune system, though the immune system will be persistent, but it is not going to work because it is not getting the feedback from the CD4 cells. Okay, so बताइए what is the difference between HIV and AIDS? It's a general awareness class. So whenever I took these classes, I generally want to know from our student whether they are being knowing what the difference between HIV and AIDS or not. Please do let me know. Anyone out of you here? Mridul, awesome queen, Jema Bharti, बताइए. सर क्वीन गई. Okay. So Mridul ji, are you there in the class? Kindly let me know what's the difference between HIV and AIDS. बताइए. Difference between HIV and AIDS. 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 AIDS and AIDS. Jema Bharti, Mridul ji, if you guys do not know, please do text over here. Sir, I do not know, please explain. But do not waste my time. If I'm asking for 2 to 3 times, at least you guys should respond कि हाँ सर आता है नहीं आता है. Such type of reluctant attitude जो होता है, it is not good for the UPSC aspirants. If you guys are knowing that, just do let me know. Yes sir, मैं इसको जानता हूँ. Otherwise, directly admit that we are not knowing it, so that it saves your own time. It saves your time also. But I want the session to be an interactive one, so that जल्दी से बताएं what the difference between HIV and AIDS. Okay, let it be. If you guys are not interested, leave this topic. Okay, so because if I'm not getting the feedback, so it's better to leave the topic of difference between HIV and AIDS. Again, always we should behave in reciprocal manner. Okay, so we have seen AIDS. In AIDS, basically the number of CD4 cell counts decreases because the virus directly attacks the CD4 cells, and because of declining of CD4 cells, what happens? Our immune system get weakened. So if you are suffering from tuberculosis or whatever it is, if your immune system is not working, so what happens? Ultimately, that person will succumb to that person जो है will die because the immune system is not working. Where is the comment, Mridul? I'm not getting your comment. I'm not getting your comment. I'm not getting your comments. I'm asking what's the difference between HIV and AIDS? डिफरेंस बिटवीन HIV एंड एड्स, चलिए बता देता हूं. डिफरेंस बिटवीन HIV एंड एड्स क्या होता है? सपोज कंसीडर टू केसेस, इट्स बेटर टू अंडरस्टैंड. ओके, चलिए कंसीडर टू केसेस. Suppose in the first cases, let us consider first case. Okay, suppose a couple is there, both had a couple is there, and apart from that, both had the sexual interaction, but both the partners were very much trustworthy. So one of the partner, whether boy or girl, he or she revealed that the person was suffering from AIDS. Okay, so what the other person will do? That after having intercourse, he directly approached the doctor. And stated all the things regarding what happened to him. So doctor said that you have to go for the ELISA test. ELISA test to get confirmed whether you have HIV or not. In the beginning, the person in the beginning itself, the doctor asks you to go for the ELISA test to check whether you are suffering from HIV or not. ठीक है ना? So what happens? He directly approached the doctor and stated all the things. The doctor said that after 1 month you can have your ELISA test, but from today itself you start your antiretroviral therapy. But from the day itself, he is on antiretroviral therapy. Retroviral therapy. ठीक है ना? The day he came to know that he had unprotected sex with someone, ठीक है ना? He went to the doctor and doctor said that after 1 month you can go for the ELISA test to check whether you are suffering from HIV or not. But from today itself you start your antiretroviral therapy. So what is the purpose of antiretroviral therapy? It prevents the virus from replication and kills the virus. What is the function of antiretroviral therapy? If the HIV virus has penetrated into the body, it replicates. If sufficient number are being found, then only it can be detected in the ELISA test. So from the day itself, he will be on antiretroviral therapy. So what happens in antiretroviral therapy? It prevents virus from replication, and also kills the virus, and also kills the virus. That means his CD4 cells are being protected. If the virus fails to replicate, and apart from that, the virus are also being killed. So ultimately, your CD4 cells are being protected. That it reflects CD4 cells are protected. ठीक है, protected. protected. So this is called HIV. This is called HIV. HIV. The person can remain in the state of HIV for 30 years. HIV, ठीक है ना? The person can remain in the state of HIV for 30 years. 30 years. If proper and on a regular basis, antiretroviral therapy are being taken care of. But in the second case, if you said, in the second case, if you see, the same two couple had intercourses, but none of them revealed the actual status. Now that person will continue to live in the same style for 2 to 3 years, and after 3 years he will go for the ELISA test. After 2 to 3 years he will go for the ELISA test. And what will happen? He will not be detected AIDS. He will not be detected AIDS. Sorry, he will not be detected HIV. Rather, he will be detected AIDS. After 3 years, after 3 years, he will be detected. After 3 years, he will be detected. He or she will be detected as AIDS because his CD4 cells count has been dropped drastically. It is around 400. So this is the difference between HIV and AIDS. Suppose the day itself he started for the precautionary measures and he will be on the antiretroviral therapy. So what will happen? The virus replication, the antiretroviral therapy stops or prevents the virus from replication. Apart from that, the virus that had been replicated, it kills them. So ultimately CD4 cells are being protected. So you can be in the state of HIV for 30 years. It will be like normal disease, just you have to be on the antiretroviral therapy on a regular basis. Apart from that, on the other case, if you see, the couple, they do not reveal to each other. They continued for 3 to 4 years. And after that, what happened? The person suddenly, when it gets weakens, went to the doctor. The doctor, after recognizing all the symptoms, he asked for ELISA test. And when the ELISA test has been conducted, he confirmed that he was suffering from AIDS because his CD4 cells has been declined drastically. It is around 400. So that's the difference between HIV and AIDS. So ultimately, you have seen that it's all about the CD4 cells, the counts. So what US patient become the first human reported to be cured of HIV after stem cell transplantation. So first of all, we must be knowing what is stem cell. Is it clear? Is it clear? Both Mridul and Jayama Bharti, is it clear to you? बताइए. Okay, not an issue. ठीक है, चलिए यहाँ फोकस कीजिए आप सभी लोग. So now we have to discuss what is stem cells. So listen, stem cells already we have discussed. What is stem cells? Basically in our body, if you see, stem cells are those special human cells that has the tendency or that has the potential to divide and differentiate into a special kind of cell. Are you getting my point? Stem cell are those special kind of cell that is having the potential or that is having the tendency to divide and differentiate into different kind of human cell. ठीक है, इसको हम stem cells कहते हैं. Just for example, when you take just for example, after sexual intercourse, the single cell that is formed is zygote है ना. The single cell, we call it zygote. And in the zygote, cell differentiation, cell division happens and it get converted into embryo. In the embryo, cell differentiation, division happens and our complete body is being developed. So you can see that zygote jo hai, it is your stem cell because it is having the potential, it is having the tendency to divide and differentiate into different kind of cell. Embryo is having when you talk about the embryo is also having the tendency, embryo is also having the potential to divide and differentiate into different kind of human cell. Even when you talk about in the bone marrow itself, in the bone marrow, we are having a stem cell called hematopoietic stem cell. In the bone marrow, we are having a stem cell called hematopoietic stem cell. That particular hematopoietic stem cell is responsible for the genesis of WBC cells, RBC cells, and platelets. So you can see that particular hematopoietic cell, it can form 3 different types of cells: WBC, RBC, and platelets. So it is also called stem cells. So when you talk about the stem cells, they are basically human cells, a special human cell that has the tendency, potential to divide and differentiate into other kind of cell. Okay. Now when you talk about stem cell classification, how it is being classified, ठीक है ना. Already we have done complete session on stem cells, so we are not going to deal completely over here, because already I have taken a special session regarding stem cell. So when you talk about stem cells, basically it is of two types. One is called somatic stem cell, also known as adult stem cell. They have the limited capacity of differentiation and may be multipotent or unipotent. So when you talk about like somatic stem cell, it is being broadly categorized into two types. One is called your somatic stem cell or adult cell, and other is called embryonic stem cell. So embryonic, when you talk about somatic stem cell, you can consider hematopoietic stem cell. You can consider hematopoietic. Example is your hematopoietic stem cell. Hematopoietic stem cell, because hematopoietic stem cell जो है, it is an adult cell. It has the limited capacity of differentiation. Maximum it can differentiate into three kind of cell. It can divide into your RBC cell, WBC cell, and your platelets. Okay, so this is called hematopoietic stem cell. Okay, so the first classification is your somatic stem cell. We call it adult stem cell. They are having the limited capacity of differentiation. One of the examples that we have cited over here is your hematopoietic stem cell. Upon differentiation division, it has the tendency to form WBC, RBC, and platelets. But when you talk about the embryonic stem cell, they are pluripotent, which means they can change into any type of cell. ठीक है ना? How? When you talk about the embryonic stem cell, the best example is your zygote. The single cell body, the single cell that is formed after the zygote. What is zygote? Single celled that is formed after fertilization. Okay, after fertilization. So this is your zygote. Now you consider this zygote. This zygote is called embryonic cell. This zygote जो है, इसको क्या कहते हैं? This zygote is called embryonic cell. Embryonic cell, because it has the tendency, it has the potential that it can form any kind of human cell in the body. This is called embryonic cell because it can form any type of cell. It can form any kind of cell, any kind of cell, because you must know that our complete body is being developed from the zygote itself, that is a single cell. That means the zygote, after cell division differentiation, it can form any kind of cell. So after cell division and differentiation, basically cell division and differentiation, it forms embryo. And already I have discussed that in embryo, basically three types of germ layers are being found. In embryo, basically three types of germ layers are being found. First layer, endoderm, mesoderm. This is called endoderm. Second layer is called mesoderm. And the outer layer is called ectoderm. Basically, if you see the endoderm is basically responsible for development of internal organs. So development of internal organ, liver, kidney, internal organs. When you talk about the mesoderm is responsible for development of connective tissue. Connective tissue मतलब होता है connective tissue in the sense blood, bone, heart, tendon, ligaments, all those things. And the ectoderm, it is responsible for nervous tissue, brain, nerve system, on all those things. So you have seen that our complete body has been started from a single cell, that is called zygote. It is called the embryonic cell because it is responsible, or it has the potential to develop any kind of cell. So there are basically two kinds of stem cells. Now what are the challenges? So you have seen that through stem cell therapy we can develop CD4 cells if its number count has been decreased. Now we have to discuss what are the challenges whenever you consider the mains paper. Often it is seen that what is the objective, what are the challenges associated with that particular technology has been often asked. So what are the challenges in application of stem cell therapy? This is much important. Okay, so we have to see challenges. So first is your safety of the patient, immune rejection of the donor cells by the host immune system post transplantation is the primary concern. So now you can see that what is the primary concern? The primary concern is safety of the patient. Now what is the safety of the patient? Suppose a patient is there. Suppose this is the patient A. This is the patient A, and he has been suffering from heart disease. Certain heart cells are being damaged. Now what is the option? The option is that he will take the heart cell from certain other person. Okay. Now the heart cell, how it will be developed? Now how heart cell will be developed? Certain, or let it take like heart disease. Now what will happen? From him, suppose a skin cell will be taken. And this skin cell, what happen? We will apply genetic engineering CRISPR-Cas9 technology. Over this skin cell, we will apply CRISPR-Cas9 technology. And we will do the genetic manipulation. And because of the genetic manipulation, this particular skin cell, it will get converted into induced pluripotent stem cell. Or you can induce pluripotent stem cell, we can call it as induced embryonic cell also. So it will get converted into induced pluripotent stem cell. Or we can also call it as embryonic stem cell. Okay, embryonic stem cells. Okay. Now suppose from skin cell we have developed the heart cell, because from the skin cell we did the genetic manipulations with the help of CRISPR-Cas9 technology, and this particular stem cell is being converted into embryonic cell. From embryonic cell we can develop the heart cell, and from this embryonic cell you can develop heart cell. Now what will happen? Now you have to introduce this heart cell over here. Now what happens? Now if this heart cell is being introduced, our immune system consider it as foreign particle. It might happen in some of the cases, it might happen that when we will introduce our own heart cell into our body, because of genetic manipulations, what happened? Our immune system, it considered this heart cell as a foreign particle and it start releasing antibodies. So because of this, the heart cell will get damaged. So that is called safety of the patient. Because whenever we are introducing anything, our immune system must accept it. If it start rejecting it and start releasing the antibodies, then it will create problem for the patient. So this is what safety of the patient. Second is your ethical concern. Use of embryo for creating human embryo, stem cells, lines may lead to commodification of human cells and tissues. Now, what is the ethical concern? You know that you might be knowing that the embryonic cell, basically there are two kinds of embryonic cell. One is a zygote and another is embryo. Both zygote and embryo, they are having the potential, they are having the tendency to that it can develop towards any kind of cell. Now, what happens? The embryo will be start selling in the market, because embryos is having the potential that it can generate any kind of cell. Now, if the things are not regulated, what will happen? Embryos will start selling. Suppose if somebody is having unwanted pregnancy, what will happen? That unwanted pregnancy will get terminated, because under that unwanted fetus or that embryo, it will start selling in the market. So commodification of human cell will start. That should not be done. And because of this only, you might be knowing that research can be conducted over the embryo, but two things must be considered. That the embryo must be the rejected one. Embryos must be rejected, but you can carry out the genetic manipulations or you can carry out the stem cell therapy, whatever it is, over the embryo, but that should not be transplanted into the uterus. It must be destroyed. So that is the ethical concern. Limited technology to generate large quantity of stem cells. Also, therapies using these avenues are largely new and much of the research and testing is needed. So limited technology, since it's a very new technology, it cannot be afforded by the common people, because equipments, machineries, skills are very less. Other concern, challenges related to gene editing modifications, potential danger of tumorigenicity. It is the tendency of cultured cell to give rise to either benign or malignant growing tumors of stem cells, possible risk of contamination and genomic changes. So what happens, it has been in research, it has been found that suppose we take certain embryonic cell and with through this embryonic cell we develop suppose heart cell and we introduce it also. So there is a possibility that our own immune system do not accept that heart cell and start having antibodies, so it gives safety for the patient. And apart from that, it is also been seen that if you introduce certain extra cells from the body, that extra cell has a tendency that it will change into tumors. It will change into tumor. So that thing should be considered. So these are the challenges. Now we have to see way forward. Now how we can overcome these challenges, way forward. So for basic clinical research and product development based on the categories of research and level of manipulation. So what thing, better regulation should be done. Government जो है better regulation should be done. कि suppose adult cell better regulation means adult stem cell therapy is being promoted. You might be knowing that hematopoietic stem cells is being permitted in India. If suppose certain person is been suffering from sickle cell disease or anemia, that person will go for the bone marrow transplantation. In bone marrow transplantation, what will happen? Hematopoietic stem cells will be injected, and that hematopoietic stem cell, it will generate new type of WBC, RBC and platelets. So this is being allowed, and apart from that, embryonic system should not be allowed. So better regulations should be promoted. Already I have stated that you can carry out stem cell therapy in an embryo, but you cannot transplant it into the uterus. Apart from that, live embryos that is there in the uterus, that should not be utilized for the purpose of research. Rather, the rejected ones should be utilized for the research. So better regulations should be done. Apart from that, informed consent for trials. Researchers should describe the risk and prospective benefits in a realistic and easily understandable manner with the potential participants. So in research, you might be knowing that in the research, few people participate as a sample, few people participate. Now, proper informed consent should be there. Apart from that, proper knowledge should be provided to the participant that these are the things which we are going to carry out. Apart from that, these are the potential dangers. So all the things should be properly stated to the participant. Scientific consideration. Appropriate measures should be taken to ensure that the stem cell derived product is safe for human application. Addressing ethical dilemma by developing guidelines like India's national guidelines for stem cell response for various stakeholders that comprehensively addresses the permissible and impermissible categories. That means proper guidelines, proper bioethical guidelines should be developed that clearly mentions the permissible area in which you can carry out the stem cell therapy. Apart from that, the non-permissible area in which stem cell therapy should not be promoted. Till date, we only promote hematopoietic stem cells that is responsible for treating the different type of blood disorders. Embryonic stem cell therapy is only for the clinical, only for the research purpose. So a proper demarcation, proper permissible and non-permissible limits should be properly demarcated. ठीक है? चलिए. Now we will see a status in India. This is of much important because often the questions are being asked regarding the regulatory system, regulatory mechanism that is being prevalent in India. So you see that when you talk about the stem cell and their derivatives fall under the definition of drugs. Remember, stem cells and their derivatives fall, and the derivatives means through stem cell different type of cells can be generated, and that different type of cells are called derivatives. Okay? So stem cells and the derivatives fall under the definition of drugs as per the Drugs and Cosmetic Act 1940 and categorized as investigational new drug, investigational new entity when used for clinical applications. So it falls under the category of drug under the Drugs and Cosmetic Act 1940. Apart from that, it is called investigational new drug. Welcome Northeast India. How are you? How are you? बताइए. Are you preparing for UPSC Northeast? Please do let me know. ठीक है. So regulated by how stem cell therapy is being regulated. So it is being regulated by National Guidelines for Stem Cell Research 2017. National Guidelines for Stem Cell Research 2017. As per these guidelines, only bone marrow and hematopoietic stem cells for blood disorder is permitted, and use of stem cells all other conditions has to be done under purview of the clinical trials in compliance with the National Guidelines for Stem Cell. See, so we are having a National Guidelines for Stem Cell Research 2017, which clearly demarcates the area in which stem cell therapy is being permitted and which clearly prohibits the area in which stem cell is not being permitted. So in the permissible limit, you might be knowing that Indian government has permitted hematopoietic stem cell for the purpose of treating blood disorder. Apart from that, the other sector, that is embryonic sector research, is being permitted only for the clinical research but not for treating the patient. So we have to follow the national guidelines. Some hospitals providing stem cell therapy in India: AIMS, New Delhi, Tata Memorial Center, and Mumbai, etc. Okay, so we have completed. Now we'll go for the genome sequencing. Is it clear to everybody? Please do let me know. I'm fine. Northeast India, are we preparing for UPSC? Please do let me know once. Just a moment. Okay, so is it clear to everybody? Now we are going to start another topic, that is called genome sequencing. So what is genome sequencing? What is the advantage of genome sequencing? All the things we have to inculcate, complete dimension. Welcome Northeast India. Northeast में कहाँ से आए? You are from which place in Northeast? बताइए, which place from Northeast? I have been to Dibrugarh, Assam, for a period of almost 14 years. Chaliye, so we will start genome sequencing in context why we are reading this, why this news has become relevant, or why. So the context is, in recently published research, an international team described the first ever sequencing of complete human genome. So Dental World, Manipur. Okay, good evening Dental World. Take it. So when you talk about the genome sequencing, what happens? For the first time, it has been found that complete human genome sequencing was being carried out for the first time. Now we will see what do we understand by human genome sequencing. Human genome sequencing क्या होता है? So when you talk about human being, almost 3.2 billion pairs of DNA is being found, and apart from that, more than 200 different types of gene is being found. Now what is genome sequencing? Suppose this is your DNA strand. Suppose this is your DNA strand. Okay, and in the DNA strand, different nitrogen bases are there. Suppose adenine is connected to thymine, thymine is connected to adenine, cytosine is connected to guanine, guanine is connected to cytosine. So this is TAGC. This TAGC basically reflects the genetic information. This TAGC, it contains, this TAGC, it reflects codon, that means genes, and it contains the genetic information. It contains the genetic information. Okay, So what is genome sequencing? Genome sequencing means basically it's a process through which the sequencing or the arrangement of nitrogen bases are being estimated. Okay, so when you talk about the genome sequencing, when you talk about genome sequencing, what it is? It is the process through which the sequences of, it is the process by which the sequences of nitrogen bases are studied. The sequences of nitrogen bases are known. ठीक है ना, are known. So this is called genome sequencing. In the basically, suppose I've already stated in the beginning itself that in human body more than 3.2 billion pairs of human DNA are being found. Apart from that, more than 20,000 type of genes are being found. Now what is genome sequencing? Basically, genome sequencing, it tries to understand the sequence of these nitrogen bases, and what does this sequence reflect? TAGC, it reflects particular type of gene, and that particular type of gene, the complete genetic information is being encoded. ठीक है ना? So now what happens in the genome sequencing? When you carry out the genome sequencing, what we will know? So suppose if complete human genome sequencing is being done, what we can found through complete human genome sequencing? Through complete human genome sequencing, through complete human genome sequencing, through complete human genome sequencing. What? First of all, if the complete human genome sequencing is being conducted, you will well acknowledge with the number of DNA pairs and apart from the number of genes. So already we know that more than 3.2 billion pairs of DNA, and apart from that more than 20,000 types of genes are being found. Apart from that, the other information you can get through the genome sequencing is your genes. Defective genes can be identified. First of all, the complete genetic informations of all the genes you can know. So complete genetic information that are found in different genes can be known. Different genes can be known. Okay. Apart from that, in the second cases, even if the genome sequencing is being conducted, you can identify the defective genes out of all the genes. So defective genes can be identified. Defective genes can be identified too. Okay. Even during the time of COVID-19, you might have recognized the regular and the continuous evolution of new variants of the coronavirus. We call it a mutation. So how do you come to know that that particular virus has mutated over here? This is because of the genome sequencing. When you found the first virus, if the first virus was being found in Wuhan, China, so the proper genome sequencing was being carried out. The proper genome sequencing means the scientist community are well aware of the different genetic information that are found in the different genes. So suppose if in certain genes genetic manipulations will be there, it will be compared to the previous one. कि यहाँ पे XYZ है, यहाँ पे now XYAA है. That means the genetic changes happen. That means it's a mutation. So it's all about your basic fundamentals regarding your genome sequencing. ठीक है? So we have discussed what is genome sequencing. Now we have to discuss the challenges associated with the genome sequencing. Challenges associated with the genome sequencing. The first challenge is your The first challenge associated is your technological issue. So like selection of analytical software tools, the speed up of the overall procedure using high performance computing technology data storage solutions. So first is what you, so genome sequencing में क्या होता है, just remember in the genome sequencing we have to do the genome sequencing of almost 3.2 billion pair of DNA. Apart from that we have to study more than 20,000 types of genes. So the data that are being generated, it is in the form of like nitrogen bases. How the data will get generated? TAGC, CA, TA, AT, GC, such type of data will be generated. Now you have to categorize this data. You have to understand the genetic information that is embedded into data. If the complete, you have to, first of all, you have to categorize this data. After that, you have to segregate this data into particular genes. You have to understand the genetic information. Out of this, again, you have to identify the defective genes. So all these things, it requires a high performance computing. So it's a very skilled work apart from the time consuming work. So high performance computing system is required. So technological issue. Apart from that, financial issue. Because of limited financial resources, the genetic projects are not as big as priority as national security and social welfare. Okay, so national security and social welfare are of much more importance. So we cannot divert our resources towards the genome sequencing. Legal issue: data privacy bill is yet to be passed. Anonymity of the data and questions of its possible use and misuse would be, suppose some person is there, he is suffering from certain disease which he do not want to know to his knowns. But if genome sequencing is being carried out, all his disease will be revealed in the particular report itself. So there is a data privacy related issue. Okay. Now what is the way forward? Because in the answer itself, if any question is being asked, you have to mention the way forward. So you can see that way forward. While India led by CSIR first sequenced an Indian genome in 2009. In 2009, it is only now its laboratory have been able to scale up whole genome sequencing and offer them to the public. Funding in the technology has been the major obstacle in scaling up the project. Funding is the major barrier. ठीक है ना? To overcome such challenges, India should go to the private funding of science projects. So private now you can carry out, suppose if you want to completely carry out the genome sequencing of your body, it can be done by the private sector itself, but it will be costing on 40 to 50,000. And 40 to 50,000, you can carry out the complete genome sequencing of the body. But you have known in India जो है, more than 90% of the people जो है, they cannot afford such technology just for knowing the defective genes. ठीक है ना? Okay. Now various initiatives taken by the genome sequencing. What are the initiatives that have been taken earlier days? So first it was a human genome project. The human genome project, it was started by USA. It was started by the USA. It was a USA project. Basically this project was being carried out to understand the genetic diversity that is being present in the US population, and India was inspired by this project, and India just started Genome India project. In this Genome India project, the Indian government जो है, it's a government project. Indian government has selected 10,000 individuals for the purpose of doing the genome sequencing. The basic objective behind this Genome India project was to recognize the genetic diversity that is existing between our Indian population. But the object was not been fulfilled. Finally, we have gone for the indigenous project. It is also in India's project. In this project, 999 people were being selected, but the selective method was awesome. Suppose if you want to select, one person was being selected from Ladakh, other person was being selected in Bihar. So to understand the genetic diversity, from the diversity of place, diversity of occupation, diversity of skills, diversity of caste system, all have been inculcated. ठीक है ना? मतलब different caste से लोगों को select किया गया, different area से लोगों को select किया गया. ठीक है? चलिए. So we have completed this also. Now we'll go for the genetically modified crops issue. Is it clear to everyone? If anybody is having the doubt, please do let me know. Please do let me know. बताइए. Any doubt up to here? So the next topic is your genetically modified crops. Genetically modified crops. You can see that recently the Department of Biotechnology, that comes under the Ministry of Science and Technology, issued guidelines for safety assessment of genomic edited plants. So it has issued guidelines regarding your safety assessment of genomic edited plants. So ठीक है ना, suppose if you want to introduce certain genetically modified crops in a particular area, whether it is going to disturb the genetic pool over there, whether it is going to disturb the indigenous plant variety, whether it is going to disturb the human gene, whether it is going to interact with the human gene. So ultimately this thing is being looked by Department of Biotechnology, that comes under the Ministry of Science and Technology. So it has issued guidelines regarding the safety assessment of genome edited plants 2002, aging norms for research into genetically modified crops. Sir, Human Genome Project is still under progress. It has been conducted. Both the project, Human Genome Project, is completed, and apart from that, Indigen Project is also completed. ठीक है ना? Yes, both the projects are being completed. Report you can study. It is available on different sites. So, first of all, we have to understand guidelines for safety assessment of genetically edited plants. So Ministry of Science and Technology, Department of Biotechnology is issued guidelines. So first of all, we have to understand what is there in the guidelines. So currently in India, all activities related to genetically engineered organism or cell and hazardous microorganisms and product there are regulated under this thing is regulated under manufacture, use, import, export and storage of hazardous microorganism, genetically engineered organism under 1989, and this thing is regulated under the Environmental Protection Act 1986. Environmental Protection Act of 1986. Okay. Another, Department of Biotechnology issued guidelines for the development and sustainable use of genome editing technologies in India, specifying the biosafety and environmental safety concern, describing the regulatory pathways to be adopted while undertaking the genome editing of plants. So Department of Biotechnology clearly mentions that if you want to introduce certain genetically modified crops, you must carry out the safety mechanism, how it is going to disturb the genetic pool, and apart from that, you must ensure that the indigenous variety of plants are not being disturbed. These are the certain criteria. Apart from that, the Genetic Engineering Appraisal Committee, the GEAC, Genetic Engineering Appraisal Committee is there, that is particularly responsible for approving the genetically modified crops. So what the Genetic Engineering Appraisal Committee do? So first of all, you must be knowing that Genetic Engineering Appraisal Committee is being established under the Environmental Protection Act 1986. Its particular objective is to approve the genetically modified crops. And how it approves? First of all, it will check whether it is going to disturb the genetic pool or not, whether it is going to disturb the indigenous variety or not. If all the things are okay, then only it will approve the particular crop. The only crop that is being approved by Genetic Engineering Appraisal Committee is your BT cotton. Now we have to study the overall policy framework for genetically modified crops. So what are the different policies that are being associated with the genetically modified crops? So first of all, rules governing the handling of genetically modified organisms and products thereof were notified in 1989 under the Environmental Protection Act 1986. Already we have discussed. Ministry of Environment, Forest and the Department of Biotechnology are responsible for implementation of this regulation. So both basically Department of Biotechnology along with the Ministry of Environment, Forest and Climate Change, they are responsible for monitoring. And apart from that, Genetic Engineering Appraisal Committee established under the Environmental Protection of 1986, it is basically responsible for approving the genetically modified crops on a commercial ground. ठीक है ना? Suppose if the genetically modified crops fails on certain ground, then it can't be approved by the Genetic Engineering Appraisal Committee. The BT cotton is the only crop that has been approved by the Genetic Engineering Appraisal Committee. genetically modified food imports require approval under the two. Suppose if you want to import something genetically modified, if you want to import genetically modified fruit crops, then it will be controlled by Environmental Protection Act 1986. Environmental Protection Act 1986. Apart from that, the Food and Safety Standard Authority Act 2006. So these are the So apart from that, when you talk about the Codex Alimentarius, Codex Alimentary Commission, it is the Joint Food and Agricultural Organization (WHO) intergovernmental body responsible for developing the standards, codes for practice, guidelines and recommendations that constitute the Codex Alimentarius, meaning the International Food Code. So Codex Alimentary is basically responsible. See, Genetic Engineering Appraisal Committee is having the authority under the Environmental Protection Act 1986 to approve or disapprove a crop, approve a genetically modified crops. But when you talk about the Codex Alimentary kernel, it can basically set a standard, it can basically set the recommendation ki suppose the following food crops should not contain genetically modified organisms, or the following crops should not contain more than this much percentage of genetically modified organisms. ठीक है ना? So such type of standard is being set by the Codex Alimentary kernel. Welcome Vivek Maurya. Codex developed principle for the human health risk analysis of GM foods in 2003. ठीक है ना? Okay. Now what is the significance of the move? What is the significance of? We have seen the Department of Biotechnology is very much concerned regarding the introduction of genetically modified crop, and apart from that, it has set the safety assessment mechanism too. Now, what is the significance? What is the significance? So you can see that aligns India regulatory framework with other mega food producing countries. So with the, now you can see that with the guidelines of safety assessment of genome edited plant that will be monitored by the Department of Biotechnology. Because of this rule, India is being aligned with the other countries. So aligns India regulatory framework with other mega food producing countries. Encourages product development and commercialization to help raise farmer income. Definitely, suppose you might be knowing that BT cotton, because of the introduction of BT cottons, we are able to generate huge amount of revenue. Okay, so it also helps to encourage the farmer income. It also helps to increase the farmer income. Speed up crop genetic development through faster breeding process. Okay, now we will see how the genetically modified crops is being done. So we'll take the example of genetically modified crops. What do you understand by genetically modified crops? We'll take the example of BT cotton to understand it. Genetically modified crops. What do you understand by genetically modified crops? Genetically modified crops. Okay, so what do you understand by genetic? Basically, when certain genes are being manipulated, or when certain gene of desired interest is being introduced into the genome of certain plants for the purpose of increasing the yield productivity, that is called genomic, that is called genetically modified crops. So what is when certain genes are being manipulated, when certain genes are being manipulated, or certain gene of desired interest, or certain genes of desired interest are being introduced in the genome of the plant, in the genome of the plant for the purpose of making it pest resistant, for the purpose of increasing the purpose being what? Objective being what? You can write the objective is called genetic objective of this. What is the objective? To make the plant pest resistant. One, to make the plant pest resistant, to make the plant pest resistant. Second is your to make the plant pest resistant. When you talk second, the second option is your to increase the yield, or productivity of the plant, to increase the yield and productivity of the plant, productivity of the plant. Okay, now we'll take the example of BT cotton. Is it clear? Vivek Maurya, Mridul, dental world. Is it clear? So we'll take the example of BT cotton, how the BT cotton is being developed. ठीक है ना? We are going to discuss BT cotton. Is it clear to everybody? So we'll take the example of BT cotton. We'll take the example of BT cotton. So you must be knowing BT cotton is the only crop that is being approved by the Genetic Engineering Appraisal Committee. So BT cotton, it's called genetically modified crops. It's the only genetically modified crops that is being approved, that is being approved by Genetic Engineering Appraisal Committee. Now you can see that, suppose this is the cotton, you might be knowing that we are cultivating the BT brinjal is not being approved. You might be knowing that we are cultivating cotton in your country from the last 5,000 to 7,000 years ago. But after 1990s, it has been found that the productivity of cotton has been decreased. It was because of the continuous infestation of pink bollworm over the bolls of the cotton. When the bolls of the cotton started to grow, it is being destroyed by the pink bollworm. So a research has been conducted in which it is being, so a research was being conducted. Suppose this is your cotton plant. This is your cotton plant and this is the soil. This is the black soil. So a research was being conducted in which it was being found that beneath the soil of the BT cotton, a bacteria was being found that is called Bacillus thuringiensis bacteria. A bacteria was being found that is called Bacillus thuringiensis. Okay, so when we did the genome sequencing of this bacteria, genome sequencing was being done, and it has been found that this bacteria DNA contains two type of gene called Cris gene. These are called Cris gene. Okay, and this Cris gene, and when this Cris gene will be introduced into the DNA of the BT cotton, when this Cris gene will be introduced into the DNA of the cotton, what will happen? It will produce certain toxin that is very much harmful to the pink bollworm. Okay, so you can see that this Cris gene it is being introduced into the DNA of, this is the DNA of the cotton plant, and this Cris gene was introduced. This Cris gene, Cris gene, Cris 1AB, Cris 1AC. So both of these genes were being introduced into the cotton seed. Now it has become a BT cotton. So how now this it has been, now listen to me again. Beneath the soil, a special type of bacteria was being found called Bacillus thuringiensis. It contains two genes, basically Cris 1AB and Cris 1AC, and both these genes, if it will be introduced into the DNA of the cotton, it will produce certain toxin, and that toxin will damage the gastrointestinal tract of your pink bollworm if it tries to taste it. Okay, so it has been inculcated into the DNA. It has been introduced in the DNA. Now it develops into the cotton plant. Now it will develop into a cotton plant. called BT cotton. Now it will call as BT cotton. And because of this, because of the introduction of these two genes, what will happen? A toxic layer will be developed. Particularly in this ball, a toxic layer will be developed. Now what will happen if the pink bollworm will come? Now if pink bollworm will try to destroy this ball, first of all he have to eat this, and when it will eat its pink bollworm gastrointestinal tract will get damaged. Pink bollworm gastrointestinal tract will get damaged. So this is how the productivity of cotton has been increased with the help of the gene that was found in the Bacillus thuringiensis. It is being introduced into the BT cotton. Is it clear to everybody? Is it clear? Please do let me know. Is it clear? बताइए. मृदुल जी, जेंटल जी, आई थिंक बीटी कॉटन इज अप्रूव्ड इन इंडिया इन 2009. देखिए बीटी कॉटन जो है, बीटी कॉटन इज बीइंग अप्रूव्ड फॉर द पर्पस ऑफ रिसर्च पर्पस. यू कैन ग्रो इट फॉर द पर्पस ऑफ रिसर्च, बट it is not being approved by the genetic engineering appraisal committee that you can grow, any farmer can grow it. Commercially it cannot be grown. ठीक है ना? So this is how. And the technology that is being employed is your recombinant DNA technology. Here recombinant DNA technology was being employed for the purpose of introducing this gene. So you can write it over here, recombinant DNA technology. What? Recombinant DNA technology was being introduced. ठीक है? Is it okay? Clear? चलिए. Now we have to discuss the most important aspect, concerns associated with the genetically modified crops. Why we have to take so much of safety concerns? Why genetic engineering appraisal committee has been responsible for approving and disapproving any of the product? So you can see that biosafety of human and animal health, risk of toxicity due to the nature of the product or changes in the metabolism and the composition of the organism resulting into the gene transfer. So you might have seen that by the introduction of BT cotton, no doubt we have increased the productivity of BT cotton. Okay, we have increased, but the pink bollworm, after a certain year, it will become extinct. Now, have you ever thought that pink bollworm is having certain linkage in the food web, is having certain linkage in the food chain? If it will get eliminated from the food web, it will get eliminated from the food chain, what will happen? It is going to disturb the food web of food chain. It is going to disturb the complete ecosystem too. Apart from that, environmental concern. Since a genetically modified organism is artificially created, its breeding with the other crops in the natural ecosystem can result in genetic contamination. So it has the tendency that genetic contamination will happen. It has the tendency that indigenous variety of crops will get impacted because of your pollination. Okay. GM technology could also allow the transfer of genes from one crop to another, creating super weeds which might be immune to common control methods. So it has the tendency that it with the help of genetic engineering, certain genetically modified crops will get created, which in the future may become weeds, and that weed you cannot control it. Or you can see that other is, suppose BT cotton is there. Pollinations happen. You might be knowing that what is pollination. Using bees, bats, they are responsible for pollination, carrying the pollen grains from one plant to another. Suppose one is your genetically modified crops and other is the indigenous variety of crop. Just consider the case. Suppose one is your genetically modified crop and other is your normal indigenous variety of crop, and the cross pollination happen. Cross pollination means the pollen grains are being transferred from the genetically modified crops to the indigenous variety. Now that particular indigenous variety will also become genetically modified crops because of cross breeding. So it has the potential that it can damage the indigenous variety of crop also. ठीक है? Okay. Economic concern. Yields of genetically modified crops as found with the BT cottons experience have turned out to be false. ठीक है ना? No doubt we have introduced the BT cottons, but the yield that was been expected in the beginning इट इज नॉट प्रोड्यूसिंग सो मच ऑफ yield, ठीक है ना। उसके बाद जो है इकोलॉजिकल कंसर्न, जेनेटिकली मॉडिफाइड क्रॉप्स could lead to erosion of biodiversity, pollute gene pools of endangered plant species. Already have discussed how the gene pools, suppose this is your genetically modified crops. Suppose this is your genetically modified crops, okay, and this is a normal indigenous variety of crop. This is your normal indigenous plant, normal indigenous plant. Now what will happen? Pollination will happen, cross pollination. This is your cross pollination, and because of cross pollination, what will happen? The pollen grains will be transferred to this indigenous variety. Now this indigenous variety, because of cross breeding, it will also become the genetically modified crops, है ना? Indigenous variety, because of cross breeding, because of cross breeding, it will also become genetically modified organisms. So genetically modified, so it has the tendency that it will damage the complete genetic pool if it is not being regulated, if it is not being controlled, if all the safety concerns are not being taken before introducing the plant. It has the tendency that it will disturb your genetic pool will get disturbed, ठीक है. Now we have to discuss the way forward. Now the way forward: awareness and information of genetically modified crops, such as keeping biosafety data out of the public domain, needs to be increased so to take the informal decisions. So first of all, we have to collect all the data, and that data should not be revealed to the public itself. It should not be revealed to the public itself. Apart from that, second is your government roadmap for ensuring food security without jeopardizing the biodiversity and compromising the safety of humans. So generally the government, what they used to take the measure, the government says that we are unable to produce so much from the indigenous variety, so it is the time that every country, they are preferring the genetically modified crops. So we have to also introduce genetically modified crop for the purpose of proper food security. But at the same time, the government should ensure that it maintains the biodiversity, it do not disturb the genetic pool. Apart from that, human health are not being concerned if they want to introduce certain genetically modified crops. So prior assessment should be carried out. Capacity building in genetically modified research agenda, development with science-based consistent. So capacity building, that means we have to carry more research so that we can get the complete data regarding the genetically modified crops. Apart from that, only such type of genetically modified crops should be introduced which do not carry the pollination process, which do not have the tendency that it will damage your genetic pool, and that maintains and conserves your biodiversity. Robust clinical trials need to be contingent or robust demonstration of safety and efficacy, adherence to the principle of Cartagena protocol on biosafety, which is an integral part of the convention of biological diversity. ठीक है? Okay? Is it okay? Clear है? Any doubt? Any doubt, friends? This is your last topic, genotransplantation. बताइए. Any doubt? Vivek Maurya, Dental World, Northeast. Please do let me know. Any doubt? If any doubt is there, please do let me know. Okay, chaliye. So now we have to study the genotransplantation. What is genotransplantation? Okay. Okay. So what is xenotransplantation? So recently doctors transplanted the heart of a genetically modified pig to a patient, highly experimental surgery to use animal organ for the life. Suppose a person was there, his heart was completely damaged. Now how to replace the heart? The heart was being replaced with the pig's heart. The heart was being replaced with the pig heart, and certain modification, certain genetic modification was being done on the pig heart. After that it was being transplanted to the human. That is called xenotransplantation. Means when animals organs are being transplanted to the human organ, it's called xenotransplantation. So pig jo hai, pig heart, it matches with the human heart. Okay, so we did it, but that experiment was not successful because the human body rejected the pig heart, and it considered the pig heart as a antigen, as a foreign material. It started releasing antibody, but the experimental was carried out. ठीक है ना? Yes, it can be called out a method. It can be, yes, dental word. ठीक है? Very good, dental. So what is xenotransplantation? It involves the transplantation, implantation and infusion of a human recipient of a live cell tissues and organs from a non-human animal. So when you talk about the xenotransplantation means the human animal organs are being transferred to the human. Animal organ are being transplanted to the human. That is called xenotransplantation. And why in this case pig was being considered? Pig are increasingly being used for this as these organs are anatomically similar to humans. The size, apart from the size, shape and everything was similar to human. That is why the pig heart was being used. Benefits क्या है? Benefits what? So what is the benefit of xenotransplantation? It could potentially provide an unlimited supply of cell, tissues, organ for human. That means we already know that in the medical world, in the medical system, just a moment. I'm sorry. So what is the benefit of xenotransplantation? Already you know in the medical world that there is a demand for human organs. Apart from that, how the demand is been increasing, we cannot afford that. That's why the scientist community of thinking of xenotransplantation through which the animal organs can be transplanted to the human. It could make it possible to alleviate graft rejection by modifying donors. But one of the greatest issue that we have recognized in this process is that whenever we try to transplant animal's organ in our body, that particular animal organs becomes a foreign particles to our body, and it start rejecting that organ by the process of releasing antibody. So what we can do, we can take the animal organ. Apart from that, with the help of CRISPR-Cas9 technology or with the help of genetic engineering, we will start the genetic manipulations. And if the genetic manipulations are being carried out properly, what will happen? It can match with the human heart. Then only that can be transplanted. Otherwise, it will act as a foreign body. Apart from that, with the help of xenotransplantation, the organ transplantation can be very much economical, can be very much cost effective. But what are the concerns? What are the concerns associated with the xenotransplantation? So first is your medical implications. Even well-matched human donor organs can be rejected after their transplantation. So we have seen that the pig heart that was been transplanted to the person was been rejected, though the pig heart was been genetically modified, manipulations are being carried out so that it can be accepted by the patient. But after 20 days or after 10 days, I do not remember, it was been rejected. It was being considered as a foreign particle, and the antibody and the human immune system started producing antibody against it. Cross-species infection of undetected and unidentified animal infection agents to patients. So now what will happen? Suppose if you are transplanting certain bodies, if you are transplanting certain organs from the animal, there is a high possibility that cross-species infection, suppose that animal was suffering from certain infection, that infection will also get transferred along with the transplantation of your particular organ. Animal rights, suppose now what will happen? The animal will be killed for the purpose of organ transplantation. Animal will be killed for the purpose of animal transplant. Though animal, they are also having the right to live. Religion, Some faith have strict rules of animals, making such transplant difficult for patients. But there are certain religions which do not allow such type of xenotransplantation. Ethical issues, difficulties of informed consent, clinical trials over humans as the technology is still emerging. Since the technology is in the emerging phase, it is very difficult to carry out the experiment, because if you want to carry out the experiment, clinical consent should be required, and the people they are not, since it's a very new phase, it is very difficult to conduct such experiment. Okay, so what is the way forward now? Xenotransplantation could be the answer of thousands of patients which will require transplant and innovative treatment for previously untreated conditions. However, there is a need to proceed with caution to ensure that safety development of this potential. So what we can do now, what is the way forward? You can carry out the xenotransplantation, but you must ensure that the organ that you are going to transplant to the other person, proper genetic manipulations, proper genetic modifications, proper genetic engineering are being employed so that they matches with the human organ. It should not be rejected, first of all. Apart from that, instead of xenotransplantation, we must focus toward the stem cell transfer. Instead of focusing towards the xenotransplantation, we can orient ourselves towards the stem cell therapy, because stem cell therapy it's also a emerging technology. But adult stem cell therapy provides adult stem cell therapy. Suppose if I am suffering from heart disease, my skin cell can be taken out, and that skin cell, certain genetic manipulations, certain genetic modifications will be carried out on the skin cell, such that it will become an induced embryonic cell, and from when it becomes an induced embryonic cell, my heart cell can be regenerated. ठीक है ना? So there is option, there is such type of option also. So I have completed my today's lecture. I hope कि आप लोगों को lecture अच्छा लगा होगा. Again tomorrow the same time we will meet for the purpose of discussing the important current affairs of science and technology. And till today, whatever the PDF is, whatever the PPTs are being made, I will just compile it into a PDF, and it will get uploaded on your Drona group. Okay. So thanks a lot everyone. Again we will meet at 10 PM sharply for the purpose of discussing the syllabus of disaster management. Okay. So thanks a lot.